Rearing of queen bees. For many years, the rearing of queen bees has occupied a key place in beekeeping. Not only the beekeeping institutes, but also many smaller beekeepers dedicate much of their work and time to this important task. Queen rearing can only be done successfully during the short period of the year when the beekeeping season reaches its peak of activity. Young queens of good origin and character are needed to form new colonies and fill winter losses. The breeding material for rearing good queens, the youngest day-old larvae, are obtained from a breeder colony. Not every beekeeper has a worthy breeder queen, but he can obtain breeding material from a beekeeping institute or from established bee breeders. The breeder colony must possess certain characteristics. It should be disinclined to swarm and have an above-average honey production potential. The bees should be gentle and not display running and bunching behavior during an inspection and show resistance to disease and parasites. The genetic endowment of the queen will therefore essentially determine the characters and quality of the progeny. The most important criterion for selection must be an above-average productivity of honey, as this factor determines the profitability of a beekeeping enterprise. The nursing colony, on the other hand, must be able to raise good queens from the breeding material introduced. It exerts no influence over the genetic character of the queen larvae it is made to foster. The nursing colony must be healthy, prolific and strong. Many bees crammed together in the smallest possible hive space is the recipe for good results when raising queens. In order to achieve the conditions, the nursing colony should have access to good pollen sources during the previous summer. When spring comes, a stimulating honey solution must be fed whenever outside nectar sources fail. All work involved must follow a timetable corresponding to the development cycle of queen bees. For grafting, the breeding material should be not older than 1.5 days. Only by divided brood care can the transfer of the started cells into another stock be effected after 24 hours. Cells are caged on the 6th or 12th day. On the 13th day, queens emerge from their cells. On the following day, mating nuclei must be formed. 3 days after that, at the earliest, mating nuclei can be sent to their mating apiaries. For precise planning, a strip with these fixed intervals for actions can be aligned with a corresponding calendar strip. Let us first turn to the work involved in preparing the breeding material. 4 or 5 days before our planned starting date for rearing queens, we remove a frame from the colony with the breeder queen. We replace it with a frame with pale comb, which had been used once only for rearing bees, and which is still moist with honey after extraction. This comb quickly attracts the queen, and the beekeeper obtains many larvae of the same age. The frame is marked with a coloured drawing pin. On the starting date, the marked comb will contain young larvae of the right age, 1 to 1.5 days old. Perfect queens can only be reared from such larvae. Here we can see the larvae literally swimming in brood food. We protect the comb from direct sunlight and against drying by wrapping it in a moistened cloth. There are many methods of preparing the breeding material for introduction to the nursing colony. One of the more simple methods is the cutting of cells. With a knife dipped in hot water, an area of comb containing larvae of suitable age is cut from the frame. On one side of the comb, all cells are shortened by 1/3 of their length. This allows bees to enlarge the mouth of the cells and draw them out as queen cells. After this, the comb is divided first into cell strips and then into pieces of comb containing individual cells. The long reverse of the cells is nipped between the halves of a split cell holder. While the method of cutting cells makes wasteful use of precious breeding material, the grafting of larvae makes more economic use of the larvae. For this method we need artificially produced cell cups. These we can easily make ourselves from beeswax. By dipping a cell mandrel of 9 mm size several times into a wax bath, held at a temperature of 65 to 70 degrees Celsius, a queen cell cup is formed at its tip. After cooling in water, the cell cup can be removed. The artificial cell cups are attached to cell holders by means of more wax, which is melted with a soldering iron. The appliance trade also offers a number of cell cups formed from plastic. The cell holders are usually attached to a cell bar by means of melted wax. We require good light, good eyesight, and a steady hand when we are grafting larvae. The young larvae are taken from their cells with a grafting tool, which is slipped under the center of the larva from the back, lifting it and transferring it. There are several methods of raising queen cells. Here we will show you the use of 3 well-known methods. For raising queen cells in a queenless colony, we need a healthy and strong colony as our nursing stock, provided with ample brood as well as nutrient. 9 days before starting, we place an additional queen excluder between the 2 stories and confine the queen to 1 hive body. 9 days later, at the start of breeding, all brood in the queenless part of the hive will be sealed. After the removal of the super and of the queen right part, in the center of the hive chamber with the sealed brood, a gap is created. Next, we form a 3-frame nucleus consisting of the old queen, 2 combs of brood, together with the adhering bees, and fill the space with a frame of stores. This nucleus is taken to an out-apiary. Into the queenless part of the colony, we brush or shake all bees from the queen-right brood chamber and also the bees out of the honey super. The nursing colony is then covered. A queen excluder at the entrance prevents strange virgin queens from entering. Bees begin to assemble in the gap. We transfer the cell-rearing frame with 20 to 30 cells into it after the colony shows the typical behavior of queenlessness. Whenever the natural flows are lacking, honey solution must be fed. The operations of this method are now shown on the stand. The nursing stock is prepared 9 days before starting. First, we find the queen and hold her temporarily in a cage. Combs with open and recently capped brood, as well as combs with pollen and honey reserves, are transferred into the lower brood chamber. The upper brood chamber holds empty combs and emerging brood.